Nirenberg and Mathieu, as well as Nirenberg and Leder's experiments, are what we're going to be talking about today. What they both did overall was help identify the codons that are now in the coding dictionary. Nirenberg and Mathieu both determined that specific RNA sequences coded for specific amino acids. And the main question that they asked was, how do these nucleotide sequences translate into amino acids? So how do these nucleotide, aka codons, translate? They used their knowledge of codons, which was provided from Brenner and Crick's experiments, to come to this question. And Brenner and Crick's experiments helped determine that codons consist of 3 non-overlapping nucleotides, which can be shown above. This is actually showing the different reading frames. However, you can see that each one is divided into 3 bases. The experiment conducted by Mathieu and Nirenberg combined poly-U chain, which was basically just a strain of U-base chain, or U-bases made into a chain, cell-free extract, which was everything but this actual cell, so it was everything in the cell basically in a tube, and then 20 amino acids, 1 being radioactive. So they completed this experiment a lot of times to come to their conclusions. What the experiment did was combine all 3 of these into a tube, add the poly-U codon. They also conducted it with a poly-A and poly-C. And then it showed that the only time that it produced a single amino acid was when it was UU, and then the third one being the U codon radioactive, following with the A and the C. It determined that UUU codes for phenylalanine, AAA codes for lysine, CCC codes for proline, and GGG was not discovered in this experiment because they could not get it to work. Nirenberg took these discoveries and then he went and collaborated with Leder, another scientist, and conducted an experiment to come to the triple binding assay. And this determined this remaining 61 codons. They asked what other matches of codons and amino acids can be made, and they used the previous findings, however, twisted it with tRNAs to identify the matches. They combined the trinucleotides, or the codons, as well as a ribosome, 20 charged tRNAs, 1 being radioactive, in a tube, and then they filtered it through a paper, and if these 3 binded, they would be stopped by the filter, and the filter would show radioactivity. However, if they did not bind, they would pass freely through. So the results, if it codes for radioactive material, then the filter shows radioactivity. And these 2 experiments are important because they led to the coding dictionary, which can be shown here. It's universal for the most part, identifies which amino acids correlate with each codon sequence. It really helped translate genetic material even further and progress the understanding of the intertwined process of codons and proteins.